the topics we're covering today, for requirements of microbial growth, we'll only cover physical and chemical factors. Because there are also biological factors. If you know, there are viruses that can kill bacteria. There are also protozoa that eats bacteria, but we won't be talking about them here. So for physical, temperature, pH and osmotic factors are important factors. Whereas for chemicals, we have carbon, nitrogen, sulfur and phosphorus. These are the major elements. Then we also have trace elements, oxygen and other organic growth factors. For temperature, you have to know the names or the terms that we use to categorize the groups of microbes based on the temperature range that they prefer. So psychrophiles are cold-loving microbes. They can grow at 0 degrees Celsius and optimum at 15 degrees Celsius. Psychotrophs also can grow at 0, but their optimum degree temperatures are higher, 20 to 30 degrees Celsius. So when you have bacteria, psychrophiles and psychotrophs, that means they pose a problem in foods that are kept in the cold. Okay, they can be a problem in the yogurt industry, in the ice cream, especially in salads. Okay, salads, because most of the salads are just washed, then they prepare and they keep in the fridge. So if the washing is and most of the vegetables are grown in soil, and soil harbors a lot of microbes. So if the washing is not done properly, then there's a chance that the microbes can be in the salads, and even though you keep in the fridge, they can still grow. They can still cause a problem. Okay? A good example would be your Listeria monocytogenes. This is one bacteria that is causing a lot of problems for salads, for coleslaw, all the kinds of foods that are kept refrigerated. Mesophiles are the moderate temperature loving microbes, grow best at 37 degrees Celsius. So most of the bacteria that you work with are mesophiles. Because the incubator that we use is often at 37 degrees Celsius. Next, we have thermophiles, the heat-loving bacteria, growth temperature optimum at 50 to 60 degrees Celsius. Where do we get this kind of bacteria, 50 to 60 degrees Celsius? We can get them from hot springs. Okay, hot springs. They thrive in hot springs. For hyperthermophiles, growth optimum of 80 degrees Celsius or higher. Usually, most of them are from the domain Archaea. Okay, Archaea. And the temperature limit for biological life now is at 122 degrees Celsius. So that is the maximum limit that they found a biological organism can thrive in. Okay, this is Archaea bacteria, Methanopyrus candelaria. Okay. So they are able to grow at 122 degrees Celsius because and the conditions are not boiling because it is under high pressure. So when it's under high pressure, water boils at a higher temperature. So even at 122 degrees Celsius, it is not boiling, and this bacteria can grow. So this is the deep sea thermal vents. Okay? Only in habitats like that, the temperatures can go above 100 degrees Celsius, and life can still thrive. if you look at this diagram, you can see the cyclophiles, cyclotrophs, mesophiles, thermophiles, and hyperthermophiles. And you can see that they actually overlap. When we grow them, we always try to grow them at their optimum temperature. But even when we grow them at the optimum temperature, don't be surprised if you get other groupings. Okay? And when you look at all these graphs, you notice that there is a minimum and a maximum. Okay? For all temperature curves, there is a minimum, maximum, and optimum. Another point to remember is the optimum is always nearer to the maximum. Okay? The optimum is always nearer to the maximum. We look at pH. So these are the examples of conditions where you can have a pH range from very acidic to very alkaline. So gastric fluids, gastric juice, lemon, vinegar, peaches, acidic soil, tomatoes. Okay. Then for neutral conditions, you have usually pure water. But once you reach seawater, it's alkaline. Okay. And the alkaline are mostly the soaps, and you have also alkaline lakes. So a bit more straightforward for pH is either acidophiles or alkaliphiles. Alkaliphiles for alkaline conditions, acidophiles for acidic conditions. Usually bacteria grow well around neutral, except for certain specific bacteria, sulfur oxidizing bacteria, for example. But most of the bacteria will grow around the neutral range. Neutral range usually from 6.5 to 7.5. Okay. When you're preparing media, don't assume that when you prepare it accordingly that the pH will be as stated by the manufacturer. If it is the first time that you are preparing that particular media, like if it's a new manufacturer, a new brand that you buy, please check the pH. Okay? Because a lot of times when you're using distilled water, double distilled water, ultra pure water, all this will affect the final pH of your product, of your agar that you're making. Okay? If it's the first time you're making it, please check the pH. For pH, usually best to remember, for molds and yeast, fungi, acidic. They prefer acidic. That's why when you look at the media for fungi, for example, potato dextrose agar, rose Bengal agar that you will be using today, they are all acidic in nature. Okay? But for acidic, you can see that the pH is usually above 5. So when you're preparing agar plates, even for fungi or mycology, you'll notice that the pH never goes below 5. Why? Because pH below 5, the agar will not harden. So you will notice that it won't harden and it doesn't work. Okay? That's why if you want to grow microbes at very acidic pH, then the best is to grow them in a broth, not agar. Okay? Not solid media, but as a broth form. Acidity is used for food reservation. So this is important. You must remember that in the past there are no refrigerators, and you need to find ways to keep food as long as as long as possible. So one of the ways was to actually pickle it. Okay, either by salts, by sugar, or by acid. Okay, But it's not by alkaline, because alkaline doesn't taste nice. Okay? So taste is also important in the food industry. Then next, we'll look at osmotic pressure. So for pH, just remember, for moles and yeast is acidic. Acidicity is also important for food preservation. Then just two categories, acidophiles and alkaliphiles, and the neutral is usually between 6.5 to 7.5. Osmotic pressure is important because most of the organisms are made up of water. You have hypertonic and hypotonic solutions. With regards to halophiles, okay, the bacteria that loves salts. Okay. So most marine bacteria are either obligate halophiles or halotolerant bacteria. So when we look at the factor salt, okay, salt. The estuary is an interesting place. So estuary is a river mouth, the place where the seawater and the river mix. So when the seawater and the river mix, there's a lot of dynamic processes that affect the salinity of the estuary. That's why in the estuary you can see categories like halotolerant bacteria. So these are usually freshwater bacteria that can tolerate salts. Then you also have facultative halophiles, okay, and also the true halophiles. Marine studies, they usually use the unit of parts per thousand to denote salinity. How much salt is in the water? So this is about 3.5%. So when you look at 20, you see here it's written 20 to 30% salt, right? So these are extreme environments that only the archaea bacteria will grow. Okay? And usually these are like salt beds, the Dead Sea, all these places where the salt in the sea, in the water is higher than normal. But then you have very interesting archaebacteria in those kind of environments. So this, I think we've already covered, just to show the effects of different osmotic solutions towards the cell. In a normal cell, in a hypertonic solution, and in a hypotonic. So hypotonic may cause a cell to burst. But for bacteria, with its cell wall, it's not a problem. It's only if you're working with eukaryotic cells without cell wall. But what you need to know is this value, sodium chloride 0.85%. That is the isotonic solution. So sometimes when you are asked to prepare diluent, right? Prepare diluent to carry out serial dilution. So it is good if you prepare isotonic solutions. Okay. So for oxygen, you need to know the names, the terms we use: obligate aerobes, obligate anaerobes, facultative anaerobes, aerotolerant anaerobes, and microaerophiles. Okay, so from the name itself, obligate means require, must have oxygen. Obligate anaerobe cannot have oxygen, because oxygen will actually kill the bacteria. Okay. Then facultative is they are able to survive both with and without oxygen. Then you have aero-tolerant anaerobes. Anaerobes, they prefer to have an anaerobic lifestyle, but they are able to survive even when exposed to oxygen. So this is important because if your anaerobe is aero-tolerant, then you can actually grow them in just normal environments, normal conditions. It's just the media that you use will have more of the reducing elements so that the bacteria can thrive better, can survive better. So if you were to prepare a soft agar here and you were to incubate it and you were to observe the growth through turbidity, you will see the pattern of growth like this. And it makes sense when you see the distribution of the bacteria based on their category to which they are affected by oxygen. So next we'll look at the chemical requirements. Carbon, nitrogen, sulfur, phosphorus, these are the main ones, these are the macronutrients. So we are all hydrocarbon based organisms, so of course we will need carbon. For nitrogen, phosphorus, if you remember your biochemistry, you can find this in your, this is mostly in ATP, in your DNA, RNA. So your nitrogen is mostly found in amino acids and also DNA, RNA. This one you can find in your two or three of your amino acids. Okay, when bacteria fixes nitrogen, the first compound that is formed is ammonia. But most of the plants and other living organisms take up nitrate. Okay? So from here itself you can know that there is a process of fixation and oxidation. So what carries out these processes? The main ones is we make fertilizers. We use industries, we use one of the chemical processes to fix nitrogen. There is also nitrogen fix during the lightning strike, because nitrogen fixation is a process that requires a lot of energy. So which makes you marvel at how bacteria can fix nitrogen at just very normal conditions. Okay, so that's the power of biological reactions. When we have phosphorus, for phosphorus, okay, most of it is from inorganic phosphate salts. Then for sulfur, also from the soil. You will find that most of the agar, or most of the media that you use to grow bacteria will have all these elements: carbon, nitrogen, sulfur, and phosphorus. Trace elements, as I mentioned, trace elements very minor amount. Okay, iron, copper, molybdenum, zinc. So the main function is as cofactors. Then other than that, organic growth compounds like other amino acids, purines, pyrimidines, and mainly vitamins. Okay? You see a lot of carbon in the form of lactose, maybe. Lactose. You see nitrogen in the form of peptone. Okay? And then phosphorus can be in the form of a buffer, phosphate buffer. And sulfur can be in the form of the ferrous sulfate. But what about the others? What about trace elements? What about growth factors? Does the bacteria not need them? Where is it added? Trace elements are so minor, normal water supply has it. So just, it's okay, it's covered. And also because most of the components that is used to make the media is not really chemical grade pure. So they will have trace elements inside. Whereas for vitamins, it is always found in yeast extract. So most of the media you'll see have the component yeast extract. So the role of the yeast extract is to provide these growth factors for the bacteria to grow. Now, most of the bacteria can synthesize their own vitamins, but not all. So when they cannot synthesize, they still have a source for their vitamins. Moisture. Moisture is present in agar. So that's the role of the agar, to trap the moisture, to provide moisture to the bacteria. It's usually prepared in 1.5 or 2%. So most of the media that is provided, when we buy it from overseas, it's usually around 1.5% agar. But if you want, you can actually add your own agar to make it 2%. Okay? Because in Malaysia, it's warmer. So if you want your agar to last longer, you can add more agar. If you want your media to last longer, you add more agar. 2% is okay. Next, light. Light is for bacteria that requires light, or microbes that require light. So all the bacteria that carries out photosynthesis will require light. Daylight lights. So when you give light, you give it 24 hours, it has a dark phase, light phase and a dark phase in photosynthesis. So the best is actually 12-hour light, 12-hour dark. Then pressure, we won't, unless you do research in deep sea microbiology, you won't be affected by pressure. Okay? Most of the time, we will be working at just normal pressure, atmospheric pressure. So pressure is more important when you're working with deep sea microbiology. So next, culture media. So these are the things that you use to grow them. So most of the time you just have to follow the manufacturer's instructions. Make sure you measure pH. And please note, not all media has to be autoclaved. Some media cannot be autoclaved. So please read the instructions properly. And some media requires supplements. So what you have to do is you read, like the usual practice is you autoclave the media. Then once the media is cooler, you add the supplements. Okay? Mix well and then you pour. So just take note that not all media is to be autoclaved, and sometimes supplements are required. Some media also require to be incubated in the dark, because sometimes they have chemicals that are light sensitive. So if you incubate it near a light source, it will degrade. Okay? Then here you see the unique properties of agar. We've already mentioned. It melts above 95 degrees Celsius, but once it melts, it will solidify at around 40 degrees Celsius and it will remain hardened. And it is also very inert. Most of the bacteria don't utilize agar, so that is very good. We want to make sure that we can control what kind of nutrients, what kind of food the bacteria is taking up. So here you can see there's agar plate, agar deep, broth and slant. Most of the time we start off with a plate. We isolate the bacteria on the plate. Then we purify the bacteria. Anything that grows, we purify them. Purify means we try to make sure that it's a single colony of the same bacteria. Because if you were to isolate from the environment, you have a mixture of bacteria. So once you purify it, you realize, but then plates don't last long. Depending on how thick you pour your plates. You can see that after 5 days, you see that the colony is still there. You see the plate still looks okay. But most probably they're all dead because there are no more nutrients. Broth also won't last. Nutrients will get up there. That's why if you want to keep your bacteria long, you prepare deep. That one easily can last 1 month. Slants also not long. So here you see the solid media I've already mentioned, 1.5 to 2%, semi-solid 0.5%, and liquids. So here you can, most of the examples given here for nutrient broth, tryptic soy broth, MRVP broth, phenolic carbohydrate broth. These are for your biochemical tests, which you will do later, not today. Then for semi-solid media, 0.5% or less, you can use it to prepare your motility agar. Now this is the soft agar. E. coli is motile, yes, then you can see non-motile, motile, and slightly, okay, less motile. You have synthetic or chemically defined media. Then you have complex media. Okay. So complex media, look at it, it has peptone, beef extract, so a lot of amino acids. So here beef extract also has the vitamins, sodium chloride, you have agar, and you have water. Here is for synthetic or chemically defined. So we know everything here, all the compounds in a chemically defined media. But for complex agar, we add peptone. But peptone can be made up of many different amino acids. We don't know what kind of amino acids is inside. We don't know the composition. We just know it's peptone. Same goes for beef extract. We don't know the exact composition. So that's the difference. For chemically defined, we know exactly what is in the media. For complex, we don't. So why do we need chemically defined media? To study biochemical processes, pathways. Okay? When you know every component in the media, then if you carry out mutagenesis to get mutants, bacterial mutants, then you can see whether the bacteria can utilize certain elements or not. The incubation that you've been carrying out until now is all aerobic, just normal atmospheric condition. Okay? Some researchers require to work with anaerobic bacteria. Anaerobic microbiology is actually quite a specialized field, okay, because a lot of things that we do normally as a microbiologist, in anaerobic microbiology you cannot do, because upon exposure to oxygen, the bacteria dies. You will notice that in all the anaerobic media that you use, there is a lot of reducing agents. Okay? Reducing agents, for example, your thioglycolate. Okay? So you also have thioglycolate, ascorbic acid, cysteine, iron fillings. This will cause the media to reduce the oxygen. So sometimes if you want, you can do this. You boil the media to force out the dissolved oxygen, and then you add a layer of oil on top to prevent oxygen from entering. So now we have oxyrase, which means in the media there is an enzyme, oxyrase, that takes up the oxygen. Essentially, the plate itself is like a anaerobic chamber like that. So you can see here, anaerobic jar. It's just like it's a jar where it's airtight. You can clamp it, and you place your plates here, and you have a gas pack. A gas pack that absorbs the oxygen, changes it to water, and releases CO2. So this will change the atmospheric condition in the anaerobic jar. When you are working with the anaerobic jar, make sure you have this palladium catalyst. Without the catalyst, the jar doesn't work. But you notice that with this anaerobic jar, you still need to open the jar, you still need to put the plates in. So for those obligate anaerobes, they can't survive. They will be exposed to air. Okay? So the ones that grow are usually microaerophiles or aerotolerant anaerobes or spore formers. But now this is the anaerobic chamber, a robust setup. You have an airlock where you place your samples in. So the samples are inside with all your media and everything. Then you change the atmosphere here, fill it up with, remove the oxygen from here before you open the chamber here and move the things from here to here. And then you lock it up again. There's an incubator here. There are all the apparatus here for you to carry out transfer and isolation. Other than that, we have enriched media and selective media. So the ones that you've been using are mostly like you use nutrient agar. That is your basic media. That's your general purpose to grow, just to grow your heterotrophs. So what about enrich? Which means we want the bacteria requires a lot of nutrients. Okay, takes up a lot of nutrients, copiotrophs. Okay. For example, blood agar. Usually blood agar is about 5%. So 5 ml in 100 ml agar to prepare blood. So 5 ml of blood in the agar. When it's cooled down, then you pour. Chocolate agar is when it's still warm, like about 60 degrees Celsius, you add the blood in. Also same 5%, that will make it cook, brown color. Then you pour. But when you add blood, you have to be careful. Because now a lot of blood has antibiotics. Okay? Even if you take blood from the cow, some of the cows have antibiotics. So you have to be very careful. So you know that there is a general purpose media, it's a basic, for example, your nutrient agar. Then you know there are enriched media for copiotrophs. You have selective and differential media. So selective media means it has components that allow you to select the bacteria that you want and prevent the bacteria that you don't want from growing. So in McConkey agar, very famous, the ones that you keep using, bile salts? It is to prevent bacteria that are not used to stay in the gastrointestinal tract from growing. So those bacteria that can grow in your gastrointestinal tract can overcome bile salts, whereas the rest that are not usually found in our gastrointestinal tract, they will be inhibited by the bile salts. So, gram-positive bacteria are usually inhibited. That's why you see most of the media that is for gastrointestinal diseases, for example, like conky, brilliant green agar, all this, they will have bile salts. Bile salts as a selective component to prevent gram-positive bacteria from growing and to allow the rest to grow, the ones that are used to growing in the gastrointestinal tract. For example, in TCBS, TCBS is thiosulfate citrate bile salt sucrose agar. So this one has bile salts also. For TCBS, it is alkaline because they want to grow marine bacteria. And as you know, we covered this just now, seawater is alkaline. Okay, so you can see that the pH here selects for marine bacteria. So in most of the media that you use and gives out different colors, you have to know what are the components that allow them to be selective and what are the components that allow them to be differential. For example, for Meconium, we said bile salts select. It is also differential because it has this combination. It has lactose and phenol ray. Lactose means it's trying to differentiate between lactose fermenter versus non-lactose fermenter, because the definition of coliform is lactose fermenter with gas. Okay, so but when the bacteria ferments lactose, it will produce acid and it will change the pH indicator, phenol red into red. That's why coliform colonies are reddish, because they ferment lactose, reduce the pH around the colony and becomes red. Okay, so this pairing makes it differential. So Meconium is both selective and differential. So most of the media, the ones that you use, the colorful ones, have both selective and differential properties. So in TCBS, I've mentioned that the pH 8 is selective, right? But pH 8 is not the only selective component. It has other components that allow it to select for the bacteria that you want. But how does it differentiate? Because it has instead of lactose, it has a sucrose. So it uses sucrose utilization to differentiate the ones that can utilize sucrose and the ones that cannot ferment sucrose. Why? Because when you're working with Vibrio, the ones that are always important is Vibrio cholera. And Vibrio cholera is the Vibrio species that can ferment sucrose, whereas most of the other Vibrios do not ferment sucrose. Okay, so other than Vibrio cholera, we also have Vibrio aeruginolyticus. So the function of the media is to tell you quickly which vibrio is interesting, which vibrio is important. So if you see yellowish colony, then you know you have to do something. You have to do another confirmatory step. For enrichment broth, there are a lot of different types, but the ones that you will most likely use: alkaline peptone water for vibrio, tetrathionite broth, selenite cysteine broth or selenite F broth, Rappaport bacillus broth. These all are for salmonella. In salmonella enrichment, these are the ones that are important. But we usually don't use this anymore. We don't use selenite cysteine because it's too toxic. Nowadays, most of the labs will use tetrathionite and Rappaport bacillus. So transport media is the media that you use to transport your sample before you go to the lab. Especially, usually swab cultures. When you swab the pus or you swab any parts of the body, then if the doctor wants a swab culture, then he'll send the swab culture over to the lab. But while transportation takes time, you do not want the swab to dry up. If it dries up, the bacteria dies. So usually there is a container where you can keep the bacteria, the swab. Okay? So there are different kinds of transport media, but it depends on what you need. If you're working with anaerobes, transport media becomes very, very critical. Okay? You must make sure your transport media supports anaerobes, which means it must have thioglycolate broth, some form of reducing agent, so that the anaerobes can survive.